Assalamualaikum and a very good evening to Sir Iqmal Hisham bin Abdul Halim, and my name is Muhammad Izzat Haikal bin Maizal. So for today, my presentations is about insulin productions in the pharmaceutical industry. So before we go more deeply to understanding about insulin production, first I will introduce you what is actually insulin. Insulin is actually a kind of peptide hormones that obtained from the beta cell and secreted inside from the pancreatic glands of the mammalian. So usually insulin play a vital role in the carbohydrate metabolism, as well as insulin hormone also help in controlling the glucose level inside the human blood, and insulin's hormone also help in treating the diabetes mellitus patients. This is because insulin hormones help to transfer the glucose from the bloodstreams to the human cells for the cellular metabolism to obtain the energy from the oxidations of the glucose. So that's why insulin hormone is very important. But the diabetes patients or the diabetic patients do not have a sufficient and enough amount of insulin inside their bloods to converting or to transferring the glucose from the bloods to their cells.
Salam and good evening. My name is Nihana Bitusa. So let's continue. The usage and application of insulin products are for diabetes mellitus patients, either type 1 or type 2 diabetes. So why insulin is important? When someone has diabetes, sometimes their pancreas doesn't make any insulin, doesn't make enough, or the insulin doesn't work properly. And that's why some people with diabetes are insulin dependent, which means they need to take it as medication, because taking insulin helps to manage their blood sugar levels. In addition, overeating and not doing enough exercise can lead to continuously high amount of sugar in the blood that can reduce the efficiency of insulin by giving it glucose than it can process.
Hi guys, I'm Nantini Selvarajin. My matrix number is 23 DBT 18F1017. Today I would like to present about the microorganism involved in the production of insulin. Come on guys, let's go into the topic. Researchers use genetic engineering to manufacture the human insulin. Without needing to depend on the animals as before, like pigs, researchers could produce the genetically engineered insulin in unlimited species now. It is now made by the genetically engineered microbes. They produce human insulin in form that is less likely cause allergic reactions and also does not contain any types of animal contaminants. Recombinant DNA is a technology that scientists developed that made it possible to insert a human gene into a genetic material of a common bacteria. The recombinant microorganism could now produce the protein encoded by the human one. Recombinant human insulin has been predominantly used in E. coli bacteria and Saccharomyces cerevisiae, mostly known as yeast, for the therapeutic use in the human. So, why E. coli? E. coli has always been preferred for the production of the recombinant insulin or other proteins, as it has other several advantages such as high growth rate, simple nutrition requirement or simple media requirement, easy to handle, high yield, and very cost effective. How about Saccharomyces cerevisiae? Saccharomyces cerevisiae is the most preferred and predominant yeast for the large scale commercial production of insulin. However, several other alternate yeast strains have been explored for insulin production. Lastly, human insulin is produced in a very controlled and a clean environment. Genetically engineered bacteria are grown in a large stainless steel, which is more known as a fermentative vessel. This vessel contains all the nutrients needed for the growth, and when the fermentation is finished or complete, the mixture containing the bacteria is harvested. It is then purified and then packaged into the bottles for the distribution process.
Assalamualaikum. My name is Nur Shafiqah. Today I want to explain about the process of producing insulin using recombinant DNA technology. They have 5 steps. 1 is the preparation. The human gene is insulated. The mRNA is taken from the cell of island of Hans. Island of Hans is the resistant of pancreas that contain endocrine cell. messenger RNA is the molecule of RNA that encodes a chemical blueprint from a protein production. The isolate gene contain the code of the human DNA, the production of insulin. The plasmid DNA of bacterial cell is taken out of the cell. Step 2 is the cutting. The plasmid DNA of the bacteria is cut out, producing plasmid ring, which is empty segment of the DNA. Resection enzyme is the enzyme that cut DNA specific nucleotide sequence known as resection site. A segment of DNA known as sticky end. Step 3 is the combination. When the plasmid ring open, the gene obtained from human cell that contain the code of protein responsibility for the production of the insulin inside into the plasmid ring, and the ring is closed. The human insulin gene is now combined when the bacteria DNA plasmid.
Hello, Assalamualaikum, and I am Nahida binti Mayazli. I want to present about continuous step on producing of insulin, which is step 4. That is, we inserted a new engineered plasmid into a bacteria, and then the resulting DNA is inserted back to a bacteria. Okay, for the 5th step, bacteria are incubated with a new recombinant plasmid, and bacteria will take up and start to produce insulin. For the conclusions, how does the resulting bacteria work? The cell need nutrient in order to grow, divide, and live. While they're live, the bacterial cell process turn on the gene for human insulin. The insulin is produced by a cell, and when the bacterial cell reproduce by dividing, the human insulin gene is also reproduced in the newly created cell. And thank you.